All right, hello everybody. This is chapter 10 and 11 in your Saladin textbook. This concerns the muscular system, so we see this conveniently flayed human here kicking a soccer ball. And unlike last time when I thought I only had the cursor as an option, now I find if I hover my cursor I can get a laser pointer, or if I go back down here again I can get a pen and draw stuff. Clumsily, it seems. I'll get better at this. So let's begin. Regarding the types of muscle tissue, you've got three, and those are, getting my laser pointer here, those are cardiac muscle, which is limited to the heart, so heart only, skeletal muscle, which are the muscles you move voluntarily with, and smooth muscle, which makes up hollow organs like your stomach, your bladder, the internal walls of your blood vessels, etc. So those are the primary types, and these are differentiated by their microscopic organization, their location, as I just mentioned, and how they are controlled by the nervous and endocrine systems. So skeletal muscle as a tissue, we're starting with skeletal here, these have long, sometimes up to 30 centimeters long, fibers, and the cells are cylindrical with many peripheral nuclei. So peripheral just means on the edge, and that is because these muscle fibers or cells are so packed with the proteins that allow contraction that the nuclei just sort of get shoved off to the edge. These do not divide. So if you are thinking about your workout habits and trying to get muscles to grow, you are performing hypertrophy, which is making existing fibers bigger, not producing new fibers where there weren't any before. You can produce new fibers, but those are produced by stem cells called myosatellite cells. So this prefix here, myo, means muscle, and satellite means orbiting something else. So myosatellite cells hang out in the neighborhood of muscles and differentiate into new muscle fibers when appropriate. The visible light and dark banding is called striations, so we refer to skeletal muscle as a striated tissue. And skeletal muscle is, of course, largely under voluntary or conscious control. So if I decide to do a motion, let's say give somebody a thumbs up, the command descends from my brain, me making a conscious decision to move down to my finger, resulting in the gesture that I intend. Now, this circled muscle right here is the striated skeletal muscle, and you might be wondering, why does each cell have multiple nuclei? Well, think about the life of a cell. All of the genetic instructions for how to make proteins live in the nucleus. If the cell is super long, having only one nucleus means that any activity that the nucleus directs has to travel potentially a very long distance to get enacted. So if I need a protein replaced at one end of a muscle cell and my nucleus is at the other end, it's going to take a lot more work to get the protein product of transcription and translation from the area near the nucleus to wherever it's supposed to go. So a way to solve that is just to have multiple nuclei situated at different lengths along the cell so that at any given point, if something needs to be replaced or dealt with, there is a nucleus nearby to deal with that. All right, so next. Nope. Okay, so characteristics of muscle tissue. Excitability. So this is one of the characteristics that we defined for life in living cells as well. In the context of muscle tissue, this means that they respond to chemical and electrical signals released from nerve cells, another excitable cell type. Conductivity refers to the ability to propagate electrical potentials over or across the membrane. So we'll go into more what electrical potentials are in a little while, but for now, think of them as having cable properties similar to an electrical wire. So if you excite one area of the membrane, that area of electrical excitation can travel over the membrane to another location. Contractility refers to the ability of muscles to shorten and generate force. Now, we're separating these two concepts because you can do different kinds of contraction. So if I am lifting, let's say, my cat, who is quite fat, off the ground, I am going to shorten my muscles to bring my arms toward my body and to lift my cat. If I am simply holding my cat against the forces of his considerable mass and gravity, I'm generating force for the same muscle length, so I'm continually generating the same amount of force to keep my cat elevated. Extensibility refers to the ability of muscles to be stretched without damaging the tissue. So before you work out, maybe you stretch, or perhaps you do so in yoga class. That just means that, similar to the contractility, the muscle can relax and even be stretched beyond relaxation without considerable damage. And elasticity refers to the fact that after you extend the muscle, it will bounce back to its original shape. So these are characteristics that are specific to muscle tissue. So when we talk about the muscular system as a system, specifically as an organ system, we're really referring only to skeletal muscle, and it makes up a huge proportion of our mass, as you can see. So we're mostly muscle. So skeletal muscle, and it also includes connective tissues making up individual muscle organs. So each muscle has a series of connective tissue wrappings that we think of as being part of the muscular systems as well. So one function is movement, of course. We can walk around, get up, get down, make mouth sounds that allow us to communicate, make face shapes that allow us to communicate, all kinds of things. Also maintenance of posture and body position, so standing up, sitting up, those kinds of things. Muscles also support soft body tissues via sphincters, controls openings and passages. They contribute to thermoregulation by either dumping heat or shivering, and you have considerable stores of fat and glycogen in and around your muscles, so there's nutrient reserves as well. If you're really hard up for nutrients, you can even break down your muscles for fuel, but this typically only happens in starvation situations. Okay, so soon in lab, you will be embarking on a journey wherein you will be expected to learn some, not all, of the 600 skeletal muscles. So this constitutes between 40 and 50% of total body weight. Obviously, this varies depending on body composition, so this is on average. Motion results from alternating contraction and relaxation of muscle cells. Muscular strength reflects the prime function of muscle. So here is one skeletal muscle cell, here is another one, here is another one, and you can see their nuclei have been pushed off to the side, just like in the illustration. These guys' prime job is to get shorter on command. To do this, energy in the form of ATP, adenosine triphosphate, our energy dollar of a cell, this contains potential energy in the form of high energy phosphate bonds. When you contract a muscle, you are converting the potential energy into kinetic or mechanical energy to generate force, perform work, whatever that looks like, and produce movement. So if you are a person who is interested in the study of muscle, how it works, how you build it, how you break it, you can become a myologist, and these people do exist. The greatest field of myology is perhaps sports medicine, because people who are athletes tend to use and strain and abuse their muscles at higher rates than the rest of us. Now, before we move on, I just want to make some points about this picture. So you'll notice that the direction of the muscle fibers is left to right across the screen. So I'm tracing it with my little laser right now. But within each fiber, there are striations, which are regular, stripy, light and dark bands that run perpendicular to the direction of the fiber. Now I want to stress this is a real scanning electron micrograph, or excuse me, not scanning electron, light microscope picture, my bad. So this is not a drawing. These striations are real. Someone made this image with a light microscope, and these striations are a consequence of the way that proteins inside of the muscle fiber are arranged, which is cool. So we don't think of proteins as being something that you can visualize very easily, but in fact, if there's enough of them in enough rows, you can totally see them. And we'll talk about what these are a little bit later. Okay, so how do you form a muscle cell? If we know that a mature muscle fiber cannot undergo mitosis, that sort of begs the question, well, how do they form in the beginning? We talked a lot about how bones form, in the skeletal system lecture, so it only makes sense for us to address this in the muscular system as well. So, during fetal development, every mature muscle fiber or cell, so note I'm using fiber and cell interchangeably, that's fairly common in anatomy. So if you hear me say muscle fiber, I mean muscle cell. These develop from 100 myoblasts, so myo, muscle, and remember an oblast is a cell that makes something. These guys fuse together. So here we have up here 3 little myoblasts. These are cells that have differentiated into a muscle cell precursor, and then these will fuse with each other to produce a larger multinucleate muscle fiber. That's why they have many nuclei. They form sort of like Voltron forms, if you're familiar with that cartoon. I'm dating myself, but it's one robot formed from a bunch of smaller robots, for those of you who have no idea what I'm talking about. So, note, one of these myoblasts does not fuse and instead sticks to the side. That's your first myosatellite cell. Here is your little baby proto-muscle fiber. So muscle growth is then a result of cellular enlargement, but not cell division. And the satellite cells will replace your muscle cells when necessary. Typically, this happens if you damage your muscle. Say you get a strain, which is torn muscle fibers. Okay, speaking of muscle fibers or cells. Recall from cell biology that we have things like the endoplasmic reticulum and the cytoplasm and the plasma membrane. Within the study of skeletal muscle, the names of things within cells are replaced with a special muscle-specific prefix so that we know that we're talking about that structure in a skeletal muscle specifically. So let me go select my pen. And I want it to be blue. Sure, why not? Okay, so this prefix that we're using that denotes muscle fiber specifically is, and here we're going to see how just terrible my handwriting is, sarco. Wow, yeah, pretty bad. So sarco is a root word that means flesh. That's a little better. So, flesh, in a context of an animal flesh that we eat, we're really eating muscle fibers, so flesh prefix applies to the skeletal muscle fibers of humans. So, note we have sarcolemma, which is the specific name for the muscle cell membrane. So it's not just a regular plasma membrane, it's a muscle cell membrane. The sarcoplasm is the cytoplasm of a skeletal muscle cell. This is special because it has large quantities of glycogen, which is a carbohydrate that's sort of like starch. It's an energy storage molecule, and filled with tiny threads called myofibrils, which these little clumps are clumps of myofibrils, and myoglobin. So myo means muscle, and a globin is a large protein. So myoglobin is sort of like hemoglobin in your blood. It's a large pigment molecule that is there to store extra oxygen in reserve. Okay, so within the skeletal muscle cell, in this picture, the sarcolemma is yellow. Have I lost my pen capabilities? Yes, I have. Like I said, still getting the hang of this. Pen. Okay, so we have transverse tubules or T-tubules. So notice that there are little holes. There's one, here's another one, here's another one, here's another one. Little holes in the surface of the relatively smooth sarcolemma that appear to go somewhere. Where they go is they form a little tunnel that descends into the interior of the muscle cell. So it gives fluid on the outside and surrounding the cell access to the internal contents of the cell via this tube network. So unfortunately, the illustrator in this choice chose to make the T-tubules on the inside purple, so here's one, here's one, here's one. The openings to them are yellow. So you can see there's a dense network of membranous tubules that goes down into and between and around the myofibrils. So when something pokes inward, we call it an invagination. So these are invaginations of the sarcolemma into the center of the cell. And these are filled with extracellular fluid. So these carry muscle action potentials down into the cell. And an action potential is a traveling area of electrical excitation. Wow, writing on a Wacom tablet while presenting is hard, you guys, but doing my best. Hopefully you can read that. So, also notice, bam, 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 there are abundant mitochondria situated between the adjacent bundles of myofibrils and throughout the cell. So these are near the muscle proteins, these little guys that use ATP during contraction. So in addition to having lots of nuclei, these have many mitochondria because their ATP demand is super high, as you might expect. If you've ever worked out and felt muscle fatigue and soreness, you can imagine that this might be true. Okay, so going back down. So muscle fibers are filled with tiny thread-like proteins called myofibrils. And those myofibrils are surrounded by networks of sarcoplasmic reticulum. So in this case, it's the blue stuff down here. So this network of mesh-like sarcoplasmic reticulum is surrounding this particular myofibril. The myofibrils themselves are made of two contractile proteins. So remember, contractile means get shorter. And of course, a protein is a chain of amino acids. So you have two kinds that interlock in something called a sarcomere, and we'll see this in a little bit. So myosin are the thick filaments. They're called that because they're literally thicker in diameter than the other filaments. So actin is the major component of the thin filaments, and these overlap with and occasionally bind to the myosin to produce contraction. So when we get to the sliding filament model, the sliding refers to the sliding of these two filaments past each other. And this is a link to a video on actin and myosin, which you are welcome to watch in your own time. Okay, sarcoplasmic reticulum. So, the sarcoplasmic reticulum is a specialized endoplasmic reticulum, specifically a smooth one. So let's write that down. Oops. Specialized smooth ER. So, if I were to ask you on a quiz or an exam, would you expect to find ribosomes on the sarcoplasmic reticulum? The answer would be no. Remember, it's a smooth ER, and the smooth ER lacks ribosomes. So this is a system of tubular sacs. So we have sacs that are divided into networks of lacy tubes, and at the end you have a big tube. And there's one big tube on either side of each section of T-tubule. So we call the big tube the terminal cisternae. Cistern A is plural. A singular one is a terminal cisterna. So these, instead of having roles in lipid production and some such, like in other regular cells, these are specifically designed to store calcium. Calcium is a cation. It comes with a charge of 2+, not just 1+, and this is in a relaxed muscle. So one of the conditions that must be met in order to accomplish muscular contraction is that calcium has to exit the sarcoplasmic reticulum to trigger muscle contraction. So one of the reasons I mentioned in the course of the skeletal system that calcium in the blood and extracellular fluid is tightly regulated is because you need calcium for muscle contraction, and that calcium ultimately, although it lives in here most of the time, comes from your extracellular fluid. So, calcium binds to a component of the myofibril and triggers a contraction. So let's go next to what exactly happens. So I mentioned when I was describing the myofibril that the thick and thin filaments overlap each other, and that happens in a pattern that creates those striations that I showed you earlier on the light micrograph. So here we can see a thick filament, and it comes in a comb-like array with other thick filaments, so it has teeth like a comb. Over here we have another comb-like array, but this is thin filaments, and the teeth overlap each other a little bit. So the I band is the region that contains only thin filaments. So it is from here over to here. And here's another one right here. The sarcomere is the area bounded by two Z discs. So I know this looks like a line, and some people even call it a line, but because this is a tubular structure, if you were to turn and look at this on his face, it would look like a corrugated disc. So the Z disc and the other Z disc are the outer boundaries of the sarcomere, so everything between two Z discs. In the overlap region, 6 thin filaments surround each thick filament. So it's hard to imagine this in this particular picture, but I'm going to draw it over here. So if you were to look at the cut face of this and zoom in on one thick filament, let's go down here, and I'm going to make my oops. There we go. So, trying to change colors there, but didn't really work out. So if I have one thick filament, it would be surrounded by 1, 2, 3, 4, 5, 6 thin ones. So when contraction occurs, all of these 6 little thin filaments are going to grab onto the thick filament, or vice versa. Okay, so myosin comprises the thick filament, which is dark fibers, so the dark bands that you saw in the microscope picture, and the actin, or thin filaments, are the light fibers. So this is a band of actin. Here we have a band of actin and myosin overlapping. This band, the A band, contains myosin and actin, and the H zone, which is here, is myosin only, no actin. There are also a variety of supporting proteins. So the Z disc is made of protein. The M line is also made of a protein. This protein is called myosin. And there's another protein called titin, which you can see down here, and you'll note that it's curly in appearance, sort of like a spring. It anchors the Z disc to the tip of the thick filament, and that just keeps the little tines of the comb from sliding off of each other. So it prevents over-relaxation or expansion of the sarcomere. So let's take a moment to look at the sarcomere in a little bit more detail, and I'm going to ask you some questions about what you think gets shorter or longer during muscular contraction or relaxation. So here we have an actual scanning electron micrograph of a skeletal muscle. So here, up here, is a little segment of nucleus. These Z discs are these very dark zones flanked by light bands. And as we established previously, the sarcomere is the area between two Z discs. The A band is a band that includes the H zone and the area of overlap where actin and myosin overlap. And as you can see here, this little pale area is the H zone where there is myosin only and no actin. So if we move these two I bands toward the M line, closer together, the I band is going to get shorter. So the distance from here to here and here to here is going to get shorter. The H zone is going to likewise get shorter, and the total sarcomere length is going to get shorter. Now, if we know that the A band is the distance from thick filament tip to thick filament tip, do you think that the A band gets shorter during contraction? So take a minute to think about that. Look at the shape of this structure and ask yourself whether you think this thing gets shorter. All right, so hopefully you've taken a moment to think about it and came up with the answer that no, the A band does not get shorter. So let's go over here and say what doesn't. So elements of the sarcomere that don't change length during contraction or relaxation. We can put A band under there. And what else can we put? Does the Z disc itself change length? Nope. Another thing that doesn't change length is the fibers themselves. So it is not actin or myosin filaments that get shorter. There is no crumpling of these things. They stay the same length. The filaments slide past each other to make the whole entire structure shorter. Okay, so let's zoom in on the sarcomere and look at the components. So we've already established that the actual proteins that make up the thick and thin filaments are myosin and actin. So in this picture, myosin is going to be this thick green one, and then actin is yellowish with some adornments, and we'll talk about what the adornments are in a little bit. So regulatory proteins are ones that control formation of cross bridges. Now, what does this mean? Let's go in here and get my pen and define it. So a cross bridge is a special link between actin and myosin. So in order for muscular contraction to happen, myosin has to grab onto the actin and yank it towards the M line, causing the filaments to slide past each other. But that can only happen under the right conditions for troponin and tropomyosin. And we'll go into more detail about this in a little while. Structural proteins are those which provide proper alignment, elasticity, and extensibility. So with respect to alignment, we've got the M line, which includes myomesin, as I mentioned previously, and the Z disc, which is made of its own stuff. So these provide the edges and middle of the sarcomere. There is also titin. You may see titin referred to as connectin. These are interchangeable terms, so you can use either, and you may see me use either. So this forms elastic filaments that link the Z disc to the thick filament to keep it on track. So you don't want this thing pivoting such that it crosses over the thin filament. It needs to stay between these two, and indeed between and at the center of the 6 actin filaments that actually surround it. Because remember, in cross-section, it actually looks like this, where M is the myosin filaments and A is the actins. So myomesin is located in the M band, and that interacts with myosin and titin. Nebulin is an actin binding protein. We're not really going to get into what nebulin does too much. Dystrophin is an important one, however. This anchors the sarcolemma to the actin specifically, not to the myosin, to the actin. And what this allows is for the sarcolemma to move with the actin filaments so that the sarcolemma doesn't get ripped during contraction. Because remember, the sarcolemma pokes into the muscle cell in the form of T-tubules. So if it is incapable of moving along with the actin, you begin to rip the sarcolemma in the area of the T-tubules, and that produces damaged muscle tissue. So if this word looks familiar to you, dystrophin, that is because it is associated with muscular dystrophy, and muscular dystrophy is an error in the gene for dystrophin, such that dystrophin is either absent or it's non-functional. So I believe there's a slide about muscular dystrophy that I included later in the presentation, so we'll talk more about that then. Okay, thick filaments. A thick filament is actually made up of many myosin molecules between 2 and 500. So these have a tail, which is long, and 2 heads, which is the business end of the myosin. So the area that actually grabs the actin is this end. So when you bundle these together, you get a bare zone in the middle with no heads, and then on either side of the M line, which would imaginarily be right here, you get this appearance where it looks like you have a big tree trunk with a lot of little globby heads poking off of it, and these are the heads that grab onto the actin during the sliding filament model of muscular contraction. Okay, so 2 intertwined actin filaments. We've got actin subunits here in red. So this is 1 globular actin unit. A row of globular G-actin makes up 1 F-actin, which is the name for this rope of G-actin, and then 2 F-actins intertwined to form the full thin filament. Each of these red globular G-actins contains an active site. So I'm going to try and grab my pen. There we go. So remember that if a protein has an active site, that means that it has enzymatic activity. So it's a place where it can interact with another protein, in this case, myosin. So it's the place where myosin can grab onto actin. However, in inactive thin filaments, the active sites form a groove that holds tropomyosin. So remember regulatory proteins, I told you I'd tell you about them later. Here we go. So tropomyosin is this long ropey guy, and it's blocking all the active sites of the G-actins that are underneath it. So that's typically between 6 and 7. And if we count, we can go 1, 2, 3, 4, 5, 6, 7. There we go. On the tropomyosin, there is a complex of 3 little proteins that together we call troponin. And these bind to calcium. So remember the calcium that we're storing in the sarcoplasmic reticulum? What does it do to allow the filaments to slide past each other? Well, calcium comes down and binds to the troponin, and when it does so, it causes a conformational change in the tropomyosin that moves the tropomyosin off of the active sites of the G-actin. So calcium binding reveals the active site so that myosin can bind to actin. Nifty, huh? So, by this regulatory function, troponin and tropomyosin act like a switch that starts and stops the shortening of a muscle cell. So if you don't have calcium on deck floating around in the sarcoplasm, the tropomyosin is always going to block the active site of the G-actin, and that precludes the ability of the myosin heads to grab the actin. Once you have calcium release, if calcium is bound to troponin, the tropomyosin will be moved off of the active site, and that facilitates myosin being able to grab onto the actin. So, other structural proteins, myosin connects titin to adjacent thick filaments, nebulin helps to align thin filaments, and dystrophin links the filaments to the sarcolemma. It also transmits tension generated to the tendon, which of course is where the muscle attaches to whatever bone it's attached to. Okay, neuromuscular junction. So I have actually produced a document that's available on D2L if you are in my class. If you are just a rando internet person, first of all, hello, why are you listening to this? Secondly, you won't have access to the document that I'm mentioning because of course you're not enrolled in my class for real. I wish you were though. So if you're one of my students, the document is under the Useful Supplements folder, and it's called Neuromuscular Junction, and it basically just takes you through the neuromuscular junction process step by step. So if you like to have a quick and dirty guide to things, that's already available to you there. So neuromuscular junction is a special synapse. Let me get my pen. And it's an area where a nerve fiber makes functional contact with the muscle. So neuro refers to nerve, and muscular refers to muscle. So we have two excitable cell types coming into contact with each other. So in the case of the neuromuscular junction, the chemical of interest is acetylcholine. So at chemical synapses, which is what this is, it's a specialized chemical synapse, you need a neurotransmitter to diffuse across a gap between two cells to accomplish transmission of a signal from this neuron into this muscle cell. And in this case, that neurotransmitter is acetylcholine, which we are going to abbreviate ACH. So, components of a neuromuscular junction. The synaptic knob is the swollen end of the nerve fiber. So if we go back here, this is a synaptic knob. Sometimes they're called an end bulb, same thing. This is a synaptic knob, this is a synaptic knob, and this is a synaptic knob. So it's a broad end piece of a neuron. The motor end plate is a special spot on the muscle cell surface that contains acetylcholine receptors on junctional folds. And I'll show you these in detail in the next slide, so just wait for that. It also contains an enzyme called acetylcholinesterase, which looks like a big, long, scary word, but if we break it down, it's a little bit easier. So acetylcholine is the neurotransmitter that diffuses from the nerve to the muscle fiber to initiate contraction. Ester is a particular organic chemistry term, so it's a group that hangs off the molecule, and ACE means enzyme. So this enzyme, acetylcholinesterase, which we can abbreviate ACHE, this breaks down leftover acetylcholine in the junction, which facilitates relaxation. So you can think of it as being the acetylcholine garbage man. It comes in and cleans up leftover acetylcholine after contraction is no longer required. The synaptic cleft is a tiny gap between nerve and muscle cells. So the synaptic end bulb's membrane is not in physical contact with the sarcolemma. There's a space. So if we were to draw that, we would say this is the syn knob or end bulb, and this is the motor end plate. Notice there's space between these two, so they're not physically touching membranes, there's a little gap. That's that cleft. Boop, throw an arrow in there. Why not? I'm getting a little bit better at this drawing stuff. So there is also a Schwann cell, which is a type of glial cell. And the Schwann cell envelops and isolates the neuromuscular junction. So it's going to wrap around the top, forming kind of a roof on this space. So this is cleft. It basically isolates the cleft such that neurotransmitter released into this cleft is trapped in there and doesn't go anywhere it's not supposed to, and the ion environment of the fluid in the cleft is tightly controlled as well to facilitate transmission of signal from here to here. Okay, So I promised you a picture of this, and here it is. We've got the motor nerve fiber, which is the axon, or transmitting part of the cell. So signal comes from this way down this way, and ultimately it comes from the brain. So let me draw a really crappy brain here. Here's our cerebrum, here's our cerebellum, here's a brainstem. Ta-da! Signal originates here and travels all the way out down through the spinal cord until it exits in a spinal nerve and eventually innervates a skeletal muscle at this spot. So signal comes down here. Here is that Schwann cell I told you about that sort of guards the cleft. Notice, as I promised, the end of the synaptic knob is not physically touching the sarcolemma. And then we have an abundance of junctional folds, which are these little guys. And notice there's a dense population of acetylcholine receptors on junctional folds themselves. So, acetylcholine receptor. Remember from cell membrane components that a receptor is a protein that binds a chemical, and that can be a neurotransmitter, it can be a hormone, but regardless of that, whatever that chemical is affects some metabolic change inside the cell without necessarily going inside of it. So ACH does not enter the skeletal muscle cell. So a great exam question that I could ask you would be to ask you true or false: during excitation of a muscle fiber, acetylcholine enters the muscle cell and binds to troponin, and hopefully you would say false, because that is total hogwash. I made it up. It's not true. So notice nearby we've got nuclei as promised because there are lots of them. We also have numerous mitochondria, especially near the sarcomere, because this is the area of the muscle fiber that's using ATP. This is a light microscopy picture of a neuromuscular junction. So notice you can see the striations, and you can see the fibers, and you can also see the axon and the neuromuscular junction itself. Pretty nifty, huh? Okay, so muscle-nerve relationship. A skeletal muscle has to be stimulated by a nerve or it will not contract. So this is one way that you can get paralysis. So if you get neurological paralysis, that means that nerve isn't talking to muscle. There's other kinds of paralysis that can occur. So if you have something wrong with the muscle fiber itself, such that it can't receive the signal from the neuron could be intact and the muscle just won't respond to it, especially if there's something getting in the way. So there's a bunch of different kinds of toxins that disrupt the neuromuscular junction specifically, and in those cases there's nothing wrong with the neuron, it just isn't capable of being stimulated because there's something in the way. So one of these is curare, which comes from South America, and it's what people use, indigenous peoples put on the tips of their arrows and blow darts to immobilize prey animals. It gets in here and binds to acetylcholine receptors so that acetylcholine cannot bind. So it blocks the ability of the neuron to talk to the muscle cell. So axons, these are the large sending ends of a neuron. Here's one. These are called somatic motor fibers. So somatic nervous system is the nervous system that innervates skeletal muscle specifically. And motor refers to muscle part. So each of these motor fibers branches into about 200 terminal branches. There's a terminal branch, there's a terminal branch, there's a terminal branch. So one signal can potentially excite up to 200 muscle fibers. Each terminal branch only supplies one muscle fiber. So none of these muscle fibers are going to get another terminal branch from another fiber. So notice in this picture here we have one axon branching into 3 terminal branches, and those branches are associated with 1, 2, 3 skeletal muscle fibers. So if I send an action potential down this way, this muscle cell is going to contract, this muscle cell is going to contract, and this muscle cell is going to contract. The ones that are in gray here are not part of this motor unit, which is all of the muscle fibers innervated by one neuron, and they will not contract. So let's discuss what a motor unit is. Et voila! A motor unit is a motor neuron and the muscle fibers it innervates. So notice from the previous slide, these were actually dispersed throughout the muscle, so they weren't in a clump. They were dispersed. So if you activate one motor unit, you'll get a weak contraction over a wide area. If you get more motor units recruited, you're going to be able to generate more force. So if I'm picking something heavy up, I'm going to generate X number of action potentials to activate X number of motor units. If somebody then sets another thing on top of the heavy thing that I'm already lifting, I'm going to activate more motor units within the same muscle to increase the amount of tension I'm generating. This is also handy because you can sustain long-term contractions as motor units take turns resting. So if one motor unit runs out of ATP, another one can take over such that you can maintain a contraction, potentially even all day. So postural control, the ability to sit upright or stand upright, is contingent upon motor units taking turns. You also have a strength versus dexterity trade-off with motor units. So small motor units contain as few as 20 muscle fibers per nerve fiber. So that's very, very small. So if you have a small number of fibers per nerve fiber, but a lot of motor units in the whole muscle, you can selectively activate only a very tiny fragment of that muscle, which gives you super fine motor control over that particular thing. So an example of this is the muscles of your eye or some of the muscles of your fingers. Strength control involves having lots and lots of muscle fibers per nerve fiber. So when you fire an action potential down one nerve fiber, you're activating a lot more skeletal muscle fibers, which generates more tension right off the bat. Okay, so generation of a synaptic transmission. First, we have an action potential, and that reaches the synaptic end bulb. Voltage-regulated calcium channels open, and calcium moves into the cell. Now I want to make sure that we note that it is the neuron specifically. So what does voltage regulated mean? This is a channel that admits only calcium, and it's only open when action potential, which is an electrical change in the cell membrane, hits it. So it's open at a particular voltage window and it's closed at all other times. So calcium gates open, calcium moves into the end of the neuron, and calcium entering the end of the neuron leads to exocytosis of synaptic vesicles. So remember, a vesicle is a little bubble of membrane inside of a cell, typically generated by the Golgi apparatus. So I'm going to draw myself a vesicle here, and then inside of this vesicle is acetylcholine. So exocytosis means that this vesicle is going to fuse with the plasma membrane at the end of the neuron, and that will blorp that acetylcholine out into the synapse or synaptic cleft. So it's going to diffuse across the synaptic cleft, and it's going to bind to an acetylcholine receptor, which is a ligand-gated sodium channel. So ligand-gated channels are channels that are only open when a particular chemical is bound to them. So for acetylcholine receptors, let's do this up here. ACH receptor is open when ACH is bound to it. Let's add a stick to this B. And closed when ACH is not. So, turns out the particular ion that the acetylcholine receptor is a gate for is sodium. So sodium rushes into the cell, and that depolarizes the membrane. So it brings the membrane from a state of electrical difference, so a lot more negative charge on the inside and a lot more positive charge on the outside, to a state that more closely resembles equilibrium. So if it was polarized before, meaning had opposite charge sides, depolarizing it means we're undoing that. So voltage-regulated sodium activation gates open. So these are sodium channels that are activated by an action potential or a local depolarization. Once you want to stop contracting, acetylcholinesterase breaks down acetylcholine, and that closes the chemically or ligand-gated sodium channels, and that stops the generation of more action potentials in the sarcolemma. So it's a little hard to talk about synaptic transmission at the neuromuscular junction since we haven't done the nervous system yet. So the next slide, we're going to summarize action potentials, but I'm also going to make a video for you guys that goes over membrane potentials, action potentials, and different kinds of channels. And that's going to be separate. I'm going to draw it all out for whatever that's worth. I'm going to try and hone my drawing skills in the meantime, but that should shed more light on what I mean by all of this, and then later you can revisit this slide and have a little better understanding of what I'm talking about. But for now, here is the action potential summarized. So, number 1 is at rest. Oops. Let's get my pen. Nope. Technical difficulties. So it seems like once I click the laser, I am not allowed to have a pen anymore unless I use my actual cursor. Okay, figured it out. So in the resting state, this is just a membrane that is waiting to depolarize, so waiting to be excited. It's not currently being excited. So this is a picture of the membrane. Here we have outside, out, and here we have inside. And we have our phospholipid bilayer, and we have our transmembrane proteins. This one is a voltage-gated sodium channel. This one is a voltage-gated potassium channel. So during a resting period for a membrane, the voltage-gated sodium and potassium channels are closed. And if you look at voltage over time, it's negative 70 millivolts is the resting potential. So resting means not doing anything. If I get this membrane to depolarize, and when we get to neurons, we'll talk more about how you do that, threshold voltage for that is negative 55. So if you depolarize this enough to get to negative 55, that will open this gate, and that lets a bunch of sodium into the cell. So we've got lots and lots of positive charge out here that's suddenly rushing in. So if the axis is millivoltage negative 70 and we're letting positive charge into the cell, the charge inside of the cell is going to increase and approach 0 because it's getting more positive in here. Turns out that this channel is voltage gated, and what that means is it only likes to be open between threshold of negative 55 and positive 30 millivolts. So that channel closes right here. A channel closes. Conversely, this channel likes to be open between positive 30 and negative 70. So it's going to let potassium out. So we just depolarize the membrane by letting a bunch of positive charge in. Now we're going to repolarize it by letting a bunch of positive charge back out. So that pushes the membrane potential down again until negative 70 is achieved once more. So one depolarization and then repolarization represents an action potential. Action potentials, though, are not limited to one membrane area, so for the sake of simplicity, we've just shown you two channels in one little chunk of membrane. What would happen in real life is this channel would open and depolarize this chunk of membrane, and that depolarization would produce enough voltage change for a neighboring sodium channel to open and let sodium in, and then its neighbor would, and so forth, such that the area of depolarization travels along the membrane from one end of a neuron to another end of a neuron. So that's how it rushes down from the brain. So, contraction of skeletal muscles. Skeletal muscles shorten because their individual sarcomeres shorten. So remember from the sarcomere slide, I gave you a list of things that do change length and things that don't, and I stressed that neither the thick or thin filaments change length during shortening, so it's not a crumpling up of the filaments. It's the overlap of fibers. So if you look at this picture, we have two sarcomeres because it's the area between two Z discs and the area between two Z discs. So if we watch, the A band is going to stay the same length, but the H zone and the I band are going to get shorter and finally almost disappear. So this is maximally contracted. So this is as contracted as you can possibly be. Note the ends of the thick filaments are almost touching the Z disc. This is partially contracted and this is relaxed. So it's the amount of overlap of the fibers that changes as sarcomeres shorten. And you can even observe this using a scanning electron micrograph. So if we look here, we've got a nice big I band, nice big H zone. Down here, little bitty H zone, little bitty I band. So this is contracted, partially contracted, totally relaxed. So, contraction of skeletal muscle requires 4 actions. First, you have to have excitation of both a nerve fiber, whoopsie, and that leads to the formation of action potentials in the muscle fiber. Excitation-contraction coupling refers to action potentials on the sarcolemma, which remember is the cell membrane of a skeletal muscle cell, activating the myofilaments. Sliding filament theory is the shortening of muscle fibers, or at least the formation of tension, via overlapping and cross-bridging of actin and myosin. And relaxation is the return of a fiber to its resting length. So I've reached just about an hour here, so I'm going to stop this PowerPoint presentation and we will talk about exactly what steps are involved in each of these in the next presentation. So thanks for watching. Sorry about the little glitches. I'm still getting the hang of this, but hopefully you find these helpful, especially if you can't make it to lecture or if you just need to review. So this is Christina Howard signing off. I will see you next time.